Now that we've had lots of practice with mitosis and we know all about the different phases and what happens in them, we can talk about a very similar process called meiosis. During this, I'm going to very quickly fly through the parts that we already know because we've had so much practice and focus on the differences that are going to occur in the different phases. Okay, but first let's start with a little pre-talk about some differences between organisms. Let's start with ways that they can reproduce. So we have asexual reproduction, which means without sex or without the exchange of genetic material. These organisms reproduce using a process that is really similar to mitosis, and all of the offspring are genetically identical. So they're little clones of one another. Or we can have organisms that reproduce using sexual reproduction, meaning they exchange genetic material. They're going to reproduce using the process of meiosis. And this provides lots of genetic variability because you're mixing genes from different organisms together. Okay, so pros and cons of each. Asexual pro, you don't have to find a mate. You decide you're ready to reproduce and bam, you just do it. Sexual, that's a con. You gotta find a mate. Asexual pro, less energy. Just takes a few hours and boom, you're done, you reproduce. Sexual, it takes a lot more energy to go through meiosis and then combine genetic material and then grow the new individual. A pro for asexual, less time. Just takes a few hours and boom, you're done. Sexual takes more time. It takes more time because you have to find a mate, and for some species that takes a really long time to find a member of the same species to mate with you. More time and energy because then you got to convince that mate to mate with you, and then it doesn't take just a few hours. It takes, for humans, 9 months to combine egg and sperm and grow a whole new individual. And then you got to care for that individual after they're born because they can't take care of themselves. But here's the big con for asexual. It provides no genetic variation. So if something comes along that can kill one of them, it can wipe out the entire species. But with sexual, that's your big pro. Lots of genetic variation. I mean, think about the bubonic plague. It wiped out a huge portion of humans. But since we have a lot of genetic variation, some people survived the bubonic plague and humans still exist. So there's a couple of different ways that asexual reproduction can happen. One is called binary fission. This is one way that bacteria or prokaryotes can reproduce, and it's really similar to mitosis. But remember, prokaryotes don't have a nucleus, so it is a little bit different, but it's pretty similar. There's also budding. This is where a small growth grows off the side and jumps off and becomes its own individual. It's asexual, so the organisms are genetically identical, but one stays big and is ready to start budding again, and the other one has to grow for a little while. This picture is an example of yeast, and you also have other organisms like hydra and coral that can do budding as well. So let's talk about our chromosomes. We have homologous chromosomes, meaning homo, the same, ologous, meaning not quite the same. Okay? Homologous chromosomes, you get 2 chromosomes, one from mom, one from dad. They carry the same genes on them. For example, you have a chromosome from mom for eye color and a chromosome from dad for eye color. But they might not be the same versions of the gene. You might have a blue-eyed gene and a green-eyed gene. Humans are more complicated than this. I'm simplifying it just to sort of get the idea of what a homologous chromosome is, okay? Pair from mom, pair from dad, but they might just have different versions of genes on them. You also have autosomes. Your autosomes are your chromosomes that are not sex chromosomes. So autosomes code for everything about you except for your secondary sex characteristics, and sex chromosomes code for your secondary sex characteristics. So this image is called a karyotype. A karyotype is a picture of someone's chromosomes when they're at their most condensed state. Okay? 1 through 22, these are your autosomes. So they code for hair color and hair type and eye color and being able to produce proteins to digest food, all that good stuff. Then the very last pair, number 23, is your sex chromosomes. This determines your secondary sex characteristics. If they're both the same size, it's XX, female. If it's XY, then you're male. So everything on here determines if you develop breasts, facial hair, testes, ovaries, etc. Those are your sex chromosomes. So meiosis. Meiosis is going to occur in sexual reproduction. You're going to take a diploid germ cell, and it's going to go through the process of meiosis, and that one diploid germ cell is going to create 4 genetically distinct haploid sperm or eggs, depending on whether you're XX or XY, okay? So the whole goal of meiosis, the whole reason why we do it, is to reduce the genetic material by half, to go from diploid to haploid. Because if we didn't do this, then you would get 2 copies of the gene from mom, 2 copies of a gene from dad, which means baby would have 4 copies of the gene. This cannot happen in animals. That's too many copies of a gene. Too many cooks in the kitchen. The soup don't turn out right. So kids don't turn out right when they have this. And by don't turn out right, I mean they never develop. It can't happen. There's too many instructions going around, and the cells never develop properly. So we've got to go haploid. 1 copy from mom, 1 copy from dad to make just the right amount in baby, 2. Okay? So cell division in meiosis is going to happen in 2 parts. Okay? We're going to divide 2 times. During mitosis, we only divided once. Now we're going to go through all the steps 2 times. Meiosis I is the reduction division. You go from 1 cell to 2 cells. Then you're going to do it all over again, meiosis II, and go from 2 cells to 4 cells, with the result being you have 4 genetically distinct cells, each with 1 copy of your genes. Okay, so let's go through meiosis I. These phases should look familiar: prophase, metaphase, anaphase, telophase, and cytokinesis. They're the same and in the same order. They're just going to be a little bit different. There's going to be some key differences. Okay, prophase I, all the same stuff is going to happen. Your chromosomes are going to condense into X shape. Your nucleus is going to start disappearing. Your centrioles are going to start migrating, and your spindle fibers are going to begin to form. All that is the exact same. Here's the difference. Our homologous chromosomes are going to pair. So the copy you got from mom and the copy you got from dad are going to pair with one another. And this is going to allow something called crossing over to occur. Crossing over is when a homologous pair, so let's say red is from mom, green is from dad, they pair up because they carry the same genes, maybe different versions of that gene, but they code for the same thing. Okay? And what they can do is they can actually cross the legs of their chromosomes and trade parts of their genetic material. So now mom's chromosome has something new. It's got a new combination here. And dad's chromosome has a little new combination here that I got from mom's. Okay, so they're actually going to switch genetic material. And the whole point of this is because it provides more genetic diversity. You get more variation in those chromosomes now because they've got new genetic material that wasn't there before. Prophase is followed by metaphase I. Okay, meta, remember, line up in the middle. So chromosomes are going to line up in the middle. The one difference is instead of 1 line of chromosomes, now our homologous chromosomes are going to line up. So the one from mom and the one from dad line up. 23 pairs, so you'll have 23 pairs in these 2 rows. And the spindle fibers are going to attach to the centromeres just like before. Okay. Now here we have something called independent assortment or random assortment. Notice that not all the blue ones are on top and all the green ones on bottom. It's totally random which one goes where. Mom's might go on top on this one, dad's might go on top on this one, dad's might go on top here, mom's on top here. It's totally random, okay? And again, that is where you get some more genetic variation. After metaphase, we have anaphase, okay? Chromosomes get pulled apart. One difference, we're not ripping apart, we're pulling apart homologous chromosomes, okay? The sister chromatids are staying connected at this point. We're not ripping apart. We're just separating the homologous chromosomes, okay? And this is where your random assortment comes in as well. They were lined up randomly, so which one goes in which direction? Also random. Anaphase is followed by telophase, and since cytokinesis is so quick, I'm going to put them on the same slide. So just like before, during telophase, your centrioles go back to looking like they're doing nothing. The nucleus starts to come back, and the cell is starting to look like a peanut or kind of like a butt. Okay? All right, now, we're not, after we pinch apart and we do cytokinesis 1, we're going to have 2 cells. But we're not going to go into interphase. Oh, no, no, no. We're in meiosis now. We're going to go right into Meiosis II. So we're going to go right into our second division. Okay? So now we need to separate the sister chromatids. Now we're going to rip them apart so we can make haploids. Okay? So here's a big picture of Meiosis II. Notice the names of the phases. They sound pretty familiar, don't they? Prophase, Metaphase, Anaphase, Telophase, and Cytokinesis. Okay, so Prophase II. Now we have 2 cells. They're going to be doing the exact same stuff. Okay, nucleus is going to disappear. Spindle fibers are going to start to appear. Centrioles are going to start migrating. Okay, now crossing over doesn't happen this time. It can't because we don't have homologous pairs anymore. Homologous pairs were already separated. Prophase goes into metaphase. Chromosomes are going to line up in the middle again. Notice we have 1 line of chromosomes again. Metaphase is followed by anaphase. Sister chromatids are ripped apart, okay? So now we're ripping apart the sister chromatids, okay? Ripping them apart. Telophase, we're back to looking like a peanut or a butt. Nucleus is starting to come back. Centrioles are back to doing nothing. Spindle fibers are gone. All that. And then we're going to do cytokinesis 2, where we just rip the cells apart. And now we have 4 cells. Okay? What's important about these 4 cells is that each one is genetically distinct due to the crossing over and due to the independent assortment. Each one is genetically different. If you don't understand this, pause and call me over. I know I went through it quickly because we've already learned all these steps and we've had so much practice. Now all you need to know is the little difference that comes in each of them. Okay, what's the reverse? So we just went through meiosis and we made eggs and we made sperms. So then it's sexual reproduction. So you got to bring them together. So you bring together a haploid sperm from somebody with chromosomes XY and a haploid egg with chromosomes XX, and you create your diploid baby. That very, very first cell that everyone starts out as is called a zygote. Okay, and then it goes and develops. So this process, putting an egg and a sperm together, is called fertilization. Okay, fertilization.